Chapter 7, A Tour of the Cell. Microscopes are a way of studying cells. There are two types of them that may ring a bell. There's light microscopes that pass light through a specimen, starting with a magnification of 100. Electron microscopes beam electrons through a specimen. Organelles and atoms, it can magnify objects as small as them. Another way of studying cells is cell fractionation. Ultracentrifuges are the best way for this to be done. They split up cells without damaging organelles and allow for their functions to be found through breaking cells down. The different types of cells is split into two, prokaryotic and eukaryotic, the latter relating to you. Eukaryotic cells are found in protist animals and plants. They compose the life of mushrooms as well as ants. They have a plethora of organelles and a nucleus. They are multicellular and are vital to the survival of us. Prokaryotic cells are only found in bacteria and archaea of all living things. Their DNA is in the nucleoid and they are unicellular beings. While they lack most organelles and are less diverse, they are still extremely abundant in the universe. The eukaryotic cell has many parts with things to be done, and each one will be explained one by one. The most important is the nucleus, carrying the majority of genes. It controls all parts of the cell and everything in between. The DNA is organized into chromatin inside, which condense into chromosomes when cells are ready to divide. Ribosomes build proteins throughout the cell. Some live free in the cytosol, while others are bound to organelles. These two types are identical and interchangeable, and the cell can adjust each's numbers to keep homeostasis stable. The endoplasmic reticulum is both rough and smooth, one with a cytoplasmic surface and the other filled with grooves. These are ribosomes that can clearly be seen, and in specialized cells, they secrete glycoproteins. Sent in transport vesicles into the Golgi apparatus, this organelle sorts, ships, and determines status. Lysosomes are membrane-bound with enzymes that are hydrolytic. Their function of digestion is very specific. They digest macromolecules best at a pH of 5. This is the perfect temperature for their functions to thrive. There are vacuoles for food and water. They can pump out excess fluid that's being a bother. Mature plants each have a central vacuole. They store compounds and keep dangerous products out of the cytosol. Mitochondria use cellular respiration to make ATP, which is used as the cell main source of energy. Chloroplasts are found in the cytoplasm roaming free and are plants' main source of energy. They use sunlight to drive synthesis with water and CO2. Without them, there wouldn't be plants for humans to use. They are made up of stroma, a part of thylakoids. Without these, chloroplasts would be an empty void. The cytoskeleton provides structural support. Some fibers contract, while others contort. There are 3 different types: microtubules, microfilaments, and intermediate filaments. They keep track of where particles are going and where they went. In plants, the cell is held together by the cell wall. It protects, maintains shape, and prevents water overhaul. The wall is split up into 3 different layers. These ensure prevention from outside dangers. Animal cells instead have an extracellular matrix linked. This makes communication between membrane and cell perfectly synced. Tight junctions connect cells with continuous belts. They fuse tightly and are unable to melt. Desosomes fasten cells together. They are reinforced by keratin, which makes communication better. Lastly, gap junctions allow small molecules to pass. These is less of a connection, but still complete certain tasks. This was Chapter 7, A Tour of the Cell. I hope you've learned something and this video has helped.